So we'll go through the introduction a little bit. Maybe we can start from you, Mike.
Mike Snyder of Stanford. I think my relation to the group is we're involved in HubMap for mapping the healthy gut, and in H10 for mapping the precancer gut from familial adenomatous polyposis. So, and like being involved in lots of other consortia as well.
Great, thank you. On those who are actively participating in this breakout session, please turn on your camera so that we can all see your face. So Joe, could you introduce yourself?
Sure. Hi, I'm Joe Ecker of Salk Institute, HHMI. I'm part of the BRAIN Initiative and also the Chan Zuckerberg Seed Network, single cells of brain, a mouse, human, part of that consortium.
I'm just reading names on the on those who have their cameras turned on. So I see Simon Gregory.
Hey, I'm a co-director of single cell sciences at Duke University, and I'm an HCA adjunct member.
Great, great. And is there anyone else who would like to turn on your camera and become active participants? Okay, there's a phone who someone calling from the phone. Of course I cannot see you, but if you are interested in, okay, we have Andrew Ade. Andrew.
Hi, I'm Andrew Ade.
Andrew Ade.
With Joe in the modality session earlier on the experimental methods. I'm an associate professor at Oregon Health and Science University, and I work. I have some funding through the BRAIN Initiative and also the Human Tumor Atlas Network.
Great, great. Anyone else who was interested in active participant? Maybe Dina, could you maybe introduce yourself?
Hi, yeah, I'm Dina Procaccini. I work at the NIH for Richard Conroy. I help him with HubMap, and I will be your scribe today. And you've probably gotten a billion emails from me in the past month. So sorry about that.
Thank you very much for organizing this.
Well, there's some pretty expert people here who I think should unmask, if you ask my opinion. But
Aviv, I see you're on. Can you?
And Anshul, I think both you guys are more talented than me, that's for sure.
I'm on. I'm also the phone.
Okay.
Because I couldn't sync the two. I have problems with my Wi-Fi. That's why I didn't also turn on the camera. So I'm both on the Zoom and on the phone.
Great, great. Thank you very much, Aviv.
I'm both things. Sorry.
Yeah. Thank you. Okay, so I presume others are here as silent observers, but I think it's also very good to have you introduce yourself briefly. Let me go through the names. We have Bob Coffey. Bob?
I'm at Vanderbilt and involved in the PCA, Precancer Atlas, and studying colorectal adenomas, sporadic colorectal adenomas.
Thank you. Apologize for if I pronounce your name incorrectly. Konstantin Krisan from UCLA. Are you able to talk? Okay, we also have Rachel Normand.
Yes, hello.
Rachel.
This is me. I'm a postdoc at the Broad Institute under Aviv Regev and Chloe Virani. I'll be working on the Human Immune Cell Atlas.
Okay, the next is, I don't know whether this is a real person or not. It says test person. I presume it's a...
I'm one of your facilitators. Not sure why it says test person. Sorry about that. My name's Costa.
Okay. Oh, Costa. Okay, of course. You're our facilitator. So great. Indeed. Thank you. And then, yeah.
Just a quick update from the other groups. One or two of them are closing out. So you may have some people coming in to join you.
Sure. Yes. Thank you.
No problem.
And then we have Anshu. Anshu, we know you, but I don't know whether everyone knows.
Yes, I'm currently in space. No, I'm just kidding. I'm assistant professor of genetics and computer science at Stanford. My lab works on machine learning methods for integrative analysis of genomic data. We've recently started working with single cell RNA and ATAC-seq, and we're interested in methods that can integrate these different modalities.
Great. Also, I think more people joined. I have Arthur Lee here. Arthur.
Hi, my name is Arthur Lee. I'm a postdoc at Boston Children's Hospital and the Broad Institute. My experience is mostly in single cell ATAC-seq, and my interest is epigenomics of developing rare cell types. Thank you.
Great, thank you. We also have Ayla Sergey.
Yes, hi, Ayala Segre. Nice to meet you. I'm an assistant professor at Mass Pioneer, Harvard Medical School. I'm part of the CZI-HCA for the Eye Cell Atlas. Josh Zanes, the lead PI, and we're kind of new to the HCA community, and in particular developing methods to integrate single cell RNA-seq expression with GWAS data, and interested in the combination of single cell with ATAC-seq, trying to identify cell type specific regulation, genetic regulation of gene expression.
Excellent. Next is Ashwari Subramanian.
Hello, everyone. I'm Aishwarya. I work with Aviv and Anna Greca at the Broad Institute, and I'm involved in the Kidney Cell Atlas efforts. Great,
thank you. And next, I would like to call on Celine Berthier.
Hi, I am Associate Research Scientist at the University of Michigan. I work with Dr. Matthias Kritker on kidney, I would say, transcriptomics, and I'm mainly working on lupus nephritis.
Great, thanks. And then next, we have Francois Agui.
Yeah, hi, I'm a scientist at the Broad Institute, working with Gary Getz and Kristen Ardley, mostly on project related to GTEx, and my main interests are genetic regulation of downstream phenotypes.
Great, thanks. We also have Meng Wang.
Hi everyone, my name is Meng Wang. I'm a postdoc in Dr. Mike Snyder's lab from Stanford. I'm interested in developing statistical methods in integrating omics data and spatial information. I'm glad to attend this meeting.
Great. I guess this earlier, okay, Roseanne Cho.
Hi, my name is Roxanne. I'm a lab technician working in Mike Snyder's lab at Stanford. And we are doing a lot of ATAC-seq and RNA-seq for our project. So I was just interested in just seeing what the current issues are and learning a little bit more about it.
Great. We also have Sarah Middleton.
Hi, I'm Sarah Middleton from GSK, just working on single cell RNA-seq mostly, and just here to listen.
Great, thanks. We have Sebastian Prezel.
Hi, I'm Sebastian. I'm from UCSD, from the Center for Epigenomics.
Great. We also have Shannon Hughes.
Hi, I'm Shannon Hughes. I'm a program director at NCI, helping to lead the HTM.
Great. And next on the list is Tarthi Malta.
Hey, I'm a young investigator at University of São Paulo, Brazil, and I'm very interested in learning more about the HCA projects.
Great, thank you. We also have Veronica Rodriguez del Blasi.
Myself. Hi, can you hear me now?
Yeah.
Yeah, so I work at Boehringer Ingelheim Pharma. We're actually quite interested in the tumor microenvironment, specifically cancer-associated fibroblasts, and of course, utilizing all the technologies and learning a lot about your current efforts, which can definitely advance the discovery aspect. Of course, also looking for key collaborators as we listen in to what your current efforts are. So I'm here more as a guest, listening and learning from all of you.
Great, thank you. I'm going along this list, and the final three are, one is Vijay Nair. Vijay? Vijay.
Hi, I'm Vijay Nair. I'm working as the lead computational bioinformatician with Dr. Matthias Kressler from University of Michigan, and mainly working in the chronic kidney disease space, especially in the diabetic kidney space, and utilizing all different modalities of data types.
Great, thank you. And when you gone? When you?
Hi, my name is Wayne Hogan. I'm a program director in NIH-HCA. Thanks.
Thank you. And last but not least, coming up is Yanxiao Zhang. Yanxiao, you probably muted. Okay, he apologized. He said his mic doesn't work. Okay, let's get started. So you probably have seen from morning session, the goal for the breakout session was to identify challenges in a subtopic area, assemble, I guess, teams of collaborators working together to address one or two challenges out of it. So that would be the goal. So we have two parts. The first part for this breakout session is to essentially discuss among ourselves, identify a set of questions that we think we could tackle. The questions would be mostly described in the form of how might we blah, blah, blah. So this would be something that we can probably try to accomplish over the next 25 to 30 minutes. And then after a short break, we'll try to review these questions and identify one or two, maybe even three, if we have time, that we can then come up with potential solutions, mostly in the form of, say, a working group or some kind of a concrete agenda to tackle such solutions. So that would be the goal for this breakout session. And I do notice that we have a huge number of experts actively working on single cell techniques and genomics or imaging based and multimodal profiling. So I'm very excited for the representation in this breakout session. And I'm hoping that we can work together to identify some of the pressing needs that we are facing and then solutions, come up with solutions in the end. So if you guys have access to Google Doc, you probably should be able to see the Google document, which is called A.A Computational Analysis or Computational Challenges in Processing Multiplex Molecular Profiles. I'll just go very briefly to introduce this topic and explain why this is some issues. Probably it doesn't require explanation, but the kind of the challenges that we can probably think about are first the breadth of the assays that we're having. What kind of biological questions do each kind of assay address? What are the most common forms of assays that, platforms of assays that are being used? And identify some of the challenges that we all face and try to solve. For example, data sparsity is a key challenge that basically casting a lot of questions about how do we extract useful information from the data we're having. And that leads to another computational challenges such as clustering cells and annotate identity of the cell clusters and give confidence on how such clustering is real or biologically relevant, and so on. I'm going to open up the platform, and I will hope that the group can probably come up with issues that you observe and you have faced, and we probably could put them on the board right now. So I would just open the platform for brainstorm and for discussions. Hi, Mike.
Well, it seems like one of the big challenges, well, first of all, I'm not sure how many people are really running multiple assays on the exact same cell, at least at omics scale. There's often smaller versions, and some people are doing this, and I'm not 100% sure how great their coverage always is. So more often people are doing different ohms on adjacent sections and then trying to integrate, so you're getting spatial information on one and mapping, say, RNA-seq or TAC-seq data on top of that. So that sort of thing. So I guess not quite sure how to break all this down, and I think that's the purpose of the 45-minute section, is do we want to take it that way, or are we taking it literally about multi-ohms on the exact same cells? That's, I guess, one question. And then second, well, I can think of many topics to cover here. One that interests me a lot, and you raised this earlier, is defining cell types, especially when you start getting high resolution with the TAC data. You can actually probably tease out even more than you would tease out with RNA-seq data. So what's a cell type, what's a cell state? And then trying to bring that into biologically interpretable domains. So I guess those are some of the questions I wouldn't mind seeing tackled.
Great, thank you. Yeah, I completely agree. These are everyday questions that we deal with. I'm sure others are seeing the same. But are there additional issues that you identify while you're dealing with the multi-model profiling data?
So hear from others. Yeah, go ahead.
Yeah.
So yeah, I mean, I think it's a really good thing to bring up, because a lot of people are doing individual modalities and then integrating those. But I'm not sure what the session structure is. So in the morning one where we were talking about multimodal tools, it was really focused on development of technologies that profile multiple properties from the same cells. But I do agree that the majority of the base of users of single cell techniques are doing single modalities for a various set of properties and then integrating those. So I'm not sure. Yeah, I think figuring out what we want to focus on makes the most sense to kick things off.
Yeah, just to give you my 2 cents, I think the challenges we face when we're dealing with multi-modalities have overlapping areas with single modality assays, but also have its unique aspect in the sense that when you deal with single modalities, you face the question, how do you integrate single cell TAC with single cell RNA? Here, in many of the assays, you actually have the data from the same cell. However, those 2 type of assays each are sparse, so they don't necessarily give you ground truth. And that is a unique challenge that we actually have to face. So I would say, if you want to ask me what should this breakout session focus on, maybe we should focus on the multi-modality integration aspect.
I'd sort of hope that we could cover the assignment of cell type and deal with cell state and overfitting the data as well. So over categorizing, because it's something that I think everyone's struggling with, is the ability to assign specific types to cells given in silico methods and available data.
That's actually a very good point. I think the issue we are dealing with multimodality profiling is does the ability to profile multiple modalities improve our ability to annotate cell types better or more accurately, right? As opposed to, say, if you have single cell attack-seq data and you cluster them, but then the question is, do they match with single cell RNA-seq clusters? It's not always easy to find a one-to-one match, right? So when you have combined attack and RNA from the same cell, then you don't have that issues or not as great as the matching issues. So that would, maybe that's one area we can basically say, can we maximize the utility of multimodality assays? So before we actually go forward, I'd like to also maybe put everyone on the same page to say what multimodal molecular profiling technologies we're dealing with. I just mentioned we have single cell attack-seq and RNA-seq combined. There are multiple methods, SkyCAR, snare-seq, and a pair-seq are the three that came into my mind, and there may be more. And then we have the assay that you can map DNA methylation in conjunction with RNA, or DNA methylation in conjunction with 3D genome architecture. You can have also tri-assays looking at RNA attack and genome sequencing from the same cells, or RNA DNA methylation and genome sequencing from the same cells. Some have a higher throughput than others, but I think the multiple modalities that we're able to profile from the same cell is rapidly increasing. Right? So you're talking about DNA, RNA, and protein. I think there are ways to profile all three from the same cell with the aid of, say, antibodies staining or something like that. So I would say, if we think about the dimensionalities we have, let's put the following multiple models: chromatin architecture, histone modification, DNA accessibility, and DNA variant, RNA abundance, and protein abundance. So those are the 6 or 7 modalities that you can imagine that can be combined and interrogated from the same time. I'm sure this has been covered in the morning session. I apologize, I was in a spatial session, so I didn't go there, but I'm sure you discussed this. So with that as the backdrop.
Well, some groups do metabolomics too, of course.
Oh yeah, definitely. Sorry, I missed that. So with this kind of as a backdrop, I guess we can discuss challenges that are associated with general common cross-platform issues. And we can also discuss challenges that are associated with specific unique technique platforms. We can probably choose to first go over challenges that are associated with everything. And I identify the cell type clustering is probably one that is going to be faced by everyone, right? So that would be one challenge, cell identification or annotation of cell identities.
Just to add to some of the co-assay spectrum, one of the things that came up in the morning session was kind of striking the balance. So if you have a co-assay that does 2 different modalities, how are you balancing between the two? At the molecular level, like in constructing these libraries, it's usually not even between them. And what's the optimal balance for that? Is it better to have really high quality for one of them and then kind of splash information on a second modality? Does that work better for integration with some, single modality data sets? It's probably going to be very different with respect to which modalities you're looking at. And then also the concept that the most common, I guess, bridge modality is typically RNA. And that's usually what guides a lot of the cell type annotation as well, just because, it's genes and that's kind of the easiest way to interpret things. So those are just some things that came up that I think are important questions.
Yeah, great. Thanks, Andrew. So we have discussed this, the type of platforms that are available or to be likely on the horizon, and also the use of the type of co-assays. Are there additional challenges that you identified or you faced in your why don't I put another topic here, which is integration. I think the question of integration comes up also in no matter what platforms you are using. For single modality assays, integration is a very big challenge, but I think it's also going to be a big challenge for multimodalities. So maybe the question is, how do we integrate data across different platforms to achieve certain biological outcomes, such as the understanding of gene regulatory networks in tissues or in different, or even in development or different scenarios. So for integration, what kind of the challenges we face? I guess, again, the question is normalizing different platforms and exchange data or putting things into a format that can be compared, and noise and sensitivity, things like that. Do people share with this concern or view on the integration?
Yeah, that's a big topic. It's one I'm supposed to chair tomorrow. So not quite sure what to say here versus tomorrow, but
oh, great.
How to integrate data is always a big issue, right?
Yes.
And the question is where it sounds like a lot of the theme here is all single cell, which is ultimately where we're trying to go. It's the theme of the meeting, but integrating bulk data with single cell data is also its own challenge.
Yeah.
Many assays are very difficult to do at a single cell level, certainly at a depth you'd like to see.
Certainly.
Since we're in the question phase, I'm not sure how deep to go into this, but yeah, I mean, I think integrating all the different types, it's a big issue. Integrating data from different kinds of assays is also always tricky. So,
Andrew brought up the idea that the RNA serves as a bridge, right? So if you're measuring proteins from, whatever level from single cells, it might not correlate so well, but if you had, if you had an RNA component to that assay, you might be able to bridge to the RNA guys, a little better, or metabolites. I don't know where that stands for single cell profiling, but if you have the RNA component in there, maybe you can bridge, because it may not necessarily translate very well depending on who you, what camp you live in about how well transcription correlates with protein levels, et cetera.
Well, for me, the biggest, there's definitely that. I think we usually try and anchor back to the spatial information. And if that happens to be protein, then we'll map RNA on the protein. We'll try and map everything back to spatial. But I recognize that when you're doing single cell RNA and single cell attack, you're probably doing those in their own, in their own right. I think the other big challenge, since we're in the question phase, probably bring this up in every session, but to me, one of the most interesting things is how to not lose the cell state information, you know, the contextual information. So you have same cell type, but given its neighborhood, potentially gives a very different state. And so that's a theme that I think is a big deal. To me, it's one of the most interesting parts about building single cell maps is the context of those cells. So measuring that when you integrate information, how do you make sure you still capture that kind of information? So that is an immune cell, an immune infiltrate into a tissue probably gives a very different signature than one that doesn't have an immune infiltrate, as an example.
Yeah. As long as we are on the integration track, I also want to point it out that integrating with, it was mentioned with with spatial resolution, that's spatial data is a big challenge. But I guess another challenge related to this is when different experiments are dealing with cells that are destroyed during the assay, then comparing between different data sets naturally will require the assumption that what you're observing as stable is being shared by samples from another setting. So the underlying assumption is how stable is that molecular modality? We know DNA is stable, right? You look at the same DNA in different cells, but how stable is RNA in terms of variability, and how variable is chromatin organization? That I guess will limit our ability to integrate. I guess what I'm trying to say is the underlying assumption associated with data integration needs to be more explicitly indicated. So in terms of computational challenges, we covered about two aspects. One is annotation, or cell clustering and annotation. And second is integration across technology platforms, including spatial platforms. Those are probably two very big topics. It also helps if we can provide more specific challenges we face and maybe make the questions more specific, more narrower than right now. But before we attempt that, can we hear a little bit more views or questions from the group who have probably thought about different things as we just laid out?
I think one of the challenges might be, this, Sebastian, might be how to deal with like millions of cells, millions of data points, and then having it not only with one modality, but have it with additional modalities without losing like the sensitivity. Because if we need to partition or subsample each of the data set, that might not lead to the resolution that we can have if we can actually handle the large data sets.
Good, good. So that's a particular aspect of the clustering, right? Where when you use certain algorithms, it's a balance between the scalability and the speed.
Yeah, and even at those scales, you know, a lot of tools are written in R, and R can't handle data sets that have, I think it's more than about 2.5 billion non-zero entries, which is pretty easy to hit once you get into, you know, larger data sets. So, you know, finding tools that can actually run some of these things has been a big challenge for my group, and that gets even bigger when you start dealing with multiple modalities.
And another would maybe be, we talked about integration of different modalities, but I think there's now like being mentioned initially, there are a lot of methods for each of the modalities actually, and how to integrate and even like how to come up with to basically account for the technical artifacts that each of these methods has and biases that might be introduced to. Especially when comparing between like data sets across multiple consortia, et cetera, and labs that generate them.
Also,
Thanks.
in addition to, in addition to assigning biological labels to individual cells, I think there's quite a bit of interest specifically towards multimodality in assigning gene labels to individual regulatory elements. And I think that's a non-trivial, problem and exercise, because even if you had perfect data, I think the framework to interpret, to attach an enhancer to a gene that it upregulates, for example, is not a trivial problem. So I think, yeah, I think that would be very useful to think about as well.
I would like to elevate that point to a more higher level. I think when you analyze multimodality data, one of the objective is to link genes to regulatory elements, and that's identified as a major objective for NIH-HCA's next 10 years research, which is to link variants to their target genes. So I think this is a very important issue that potentially this working group is uniquely positioned to address. I'd like to also elaborate with probably what Sebastian talked about. The technical challenges that we face is the potential for mislabeling of cells. Example is when you do job sequencing, you have doublets, and different platforms have different rates of doublets being present. Combinatorial barcoding also have the same issue. This is called barcode collision. And how to identify the collided barcodes and how to remove such noise is, again, something we face all the time. I guess I mean, are there other topics that we have? And
well, it's confusing. Go ahead.
Just often when you're looking at multimodal assays, typically the analysis goes more in the form of you analyze the cells in the context of, say, their RNA component. Then you analyze them in the context of their attack component. There aren't a lot of tools out there, at least that I've found, that really simultaneously look at both properties at the same time to try and jointly learn what the landscape is. So I think in papers I've read, it's mostly been each of them separately and then bringing them together, as opposed to starting at the beginning with both modalities, which seems like it would be the best way.
No, I like that. That's a good topic. Trying to get more value out of, maybe that's a general theme, how to better enhance information we can get out of multimodality.
Okay, I'm not sure we captured that. Andrew, can you say again?
Yeah, just tools that simultaneously take into account information from multiple modalities, as opposed to focusing on one or the other and then integrating.
Yes.
Yeah,
I agree with Andrew.
Yeah, you can increase your value if you were to combine the data and analyze it versus analyzing it separately.
Yes,
That's
I
a good
agree.
topic. I don't think that's well taken on. I like that.
Yeah, it's true. It's multimodalities tend to talk better together if you analyze them together instead of sequentially.
Mm-hmm.
Yeah. So what have been tried in this space? Andrew, can you, do you have any information on that?
I mean, you know, we typically do the route where we analyze each separately. We've tried some like factorization-based approaches, and then, you know, for each modality and integrating the factors to try and analyze them at the same time. You know, my lab, we don't really develop these, you know, algorithms. So hopefully someone much more skilled in that space than I will take on these challenges. But I think there's definitely a need, because we're typically just going the route where we analyze them separately and integrate as early as we can, but I don't think it's as early as we would like. I mean, ideally from the get-go, you're doing this kind of simultaneous analysis, maybe even from raw reads before you even do like peak calling or you know, transcript annotation and quantification, but actual, like, you know, going as early as you can, taking into account multiple modalities.
During the break, I try to, if you open up the Google Doc, it's called 8A, Computational Challenges. If you can open this Google Doc, I took the liberty to summarize some of the key questions that were discussed in the morning, sorry, not in the morning, earlier in the first session. And there, I captured 5, and let me read them. And if you think that I missed 1 or 2 points, or please do say. First one is, how might we simultaneously taking information from multiple modalities and combine them and analyze them together? So that was a challenge we face. As a second, we identify as the challenges, how do we balance sensitivity of data processing with scalability of the algorithm, for example, dealing with millions of cells without losing the sensitivity of capturing cell types. Third is, how do we define cell types when you have high resolution data, when you essentially bring them into interpretable biological entity. And fourth is, how do I assign distal elements to target genes using multimodal profiles? And finally, how do we account for noises in the data sets, such as barcode collision and so on? So those are the 5 main challenges that I captured. Did I miss anything? Can you, this is the time that we can speak up, and then we can discuss among ourselves, maybe prioritize this, and then try to lay out some plans to move forward. Our goal is to, if you look at the blue box, our goal is to at least populate 1 such blue box, and maybe more if we have time. So do you all agree with the list of questions that I posed?
Yes.
Yeah, it looks fine.
Yeah, it looks good to me. I'm just wondering whether the data noise or missingness should be first. So to address those before we talk about data integration.
This is, again, I'm glad you agree with that. So we could, of course, prioritize this after we'll just have a discussion on how to prioritize them. But I just want to first make sure that I captured all the essential questions we posed in the earlier session. If you don't see I'm missing anything, what I then would like to do is to have some input from this group, from the participants, regarding what questions among these 5 that we can attempt to address or to tackle together by the formation of a, say, working group after this breakout session. So what challenges is most important, or that you see most urgent, most pressing, and probably solvable? or probably require a collaborative effort. So can maybe, if I go through this list from 1 to 5, can you maybe, well, we'll just take a voice vote to say whether this is we can focus on. First, how might we simultaneously taking information from multiple modalities and analyze them together? Is that a big challenge for? Yeah, I see one. You can say, I couldn't tell actually right now. You can shout maybe.
Yeah, I don't know any, I don't really know many computational approaches to solve that problem at this point. Usually it's exactly what Andrew said, was do a multimodal assay and then take and analyze each of those separately and then try to merge that back since you know what cell it's from.
At a single cell level, that's true.
Yeah.
So basically, this is a problem that certainly without a good answer right now, right? And so I hear.
Is that a collaborative project, or is that a project that one group just needs, or a couple groups need to take the lead on? Item 3 is clearly a very collaborative project. You have to agree on that across consortia if you ever want to compare data.
Yeah, I mean, I could see item 1, being just something that groups that are already trying to tackle data integration start really diving into that space. I know we had Anshul on here before, but I don't know if he's in the room anymore. But groups like that do a lot of this analysis could probably tackle that. So I'm not sure if it needs a consortium wide.
Well, we have some opinions on this now. Let's move to the second, how to deal with scalability and sensitivity issues. That probably fall in the same category as the first one, right? I guess we all have our own algorithms and how to balance this. I guess the third one, the defining cell types and bring the biology, align the computational in silico annotation to the real world physiological context. That seemed to be a big challenge.
Yeah, and that's a real single cell mapping problem, something everybody deals with, right?
Mm-hmm.
Pretty fundamental.
So let me move that to maybe
the blue box
first. Okay, let's move that to blue box. Good point.
I don't know. You lost something now.
I didn't. Did I? Okay.
Oh, there it is. It's just combined with 3. I think.
Yes.
Oh no, it isn't. I don't know. There was a 4th there. Okay.
Yes, it's fine. It's fine.
Yeah, number 4 is a real encode kind of project, but
well,
number
I think
4
you moved
is
it to the blue box, so I think we're all right.
We're all right. Yes.
Yeah.
I didn't lose it. I moved it to first, but so assign distal element to multimodal. Targeting. That's Mike. You think that's not something that we should be bothered about here, right?
Well, I guess it's something we all have to use, but it's sort of like what was the first one, wherever it is now. Yeah, I think some groups, there are different approaches for this already, as you well know. Is that something that as a collaborative type of, I guess we all need to, it'd be nice if we all use the same method.
Mm-hmm.
I don't know that it needs a consortium-wide effort to come up with the tools for that, but I may be wrong.
Mm-hmm. Okay. And then the last question I posed was how to account for noises in the data sets. I guess that's something that we'll have to each one of us face, but it's platform specific.
Yeah, also another component of the noise beyond just like cell and barcode collisions is, this was brought up in the morning session, but like contamination across cells. So, you know, quantifying how much ambient chromatin that sticks to other nuclei or things like that. And
yeah.
You know, that's a real challenge, particularly dealing with actual tissues as opposed to just doing barnyards on cell lines. So that's another noise that I think is really important to take into account. And using multimodal data could help actually with that, because those will likely have two different kind of rates of how much that molecular property bleeds into other cells based on just the platform that's being used.
Do you think that's a computational challenge or experimental challenge or both?
It's both, I think. I think, you know, ideally reducing it as much as possible on the experimental end, but there's always going to be some amount with pretty much any platform that you're going to be working with. So identifying that computationally, at least quantifying it, and maybe knowing how to take that into account in your analysis.
Okay. All right. How many people felt excited about this topic? Oh, I see a hand waving. So can we maybe move this to a second blue box?
Yeah, it's probably one everybody has to deal with.
Yeah.
I don't know if it's a topic of other sessions or not. I haven't looked at all the sessions.
Okay. Well, let's put it there, and I will try, if we have time after we deal with the first one, we'll try to deal with this one. Okay, very good, very good. So in the remaining time, we could probably try to describe this problem more specifically and come up with some steps that we can do to address them, and form a working group, identify mechanism for this to work. So now if you go to the Google Doc, there's a blue box, and I copy and paste this question there. It's a very crude phrasing right now, how to define cell types when you get high resolution data. Perhaps what we probably want to be more specific are what kind of data that would be used to serve such purposes. Let me try to put them down here. So of course, single cell RNA seq / attack seq would be the type of data. But in the same time, we also have single cell methyl C or single nucleon seq type of assays. We know RNA is generally used to define cell types, but open chromatin, DNA methylation also have similar use and utility, and it's arguable which one is better. And if you have spatial transcriptomics, for example, MERFISH data, the location of the cells and the shape and morphology also can confer information about cell types. And also antibody-based approach, such as CyTOF, could also serve this purpose.
Also, Bing, we you know, as you know, and you have been also a paper on this, is that you can use the structural information, the looping information, to actually identify cell types that you need.
That's right. The 3D conformation of the genome definitely has information on cell type specificity. How to use that, I think it's a very interesting question to address. Okay, so that currently are the data sets that we are dealing with. And each one has its own pros and cons. From my own experience, I think RNA certainly gave pretty good ability to annotate cell types, because mostly the cell types are defined by the marker genes. Right? Traditionally, the histology is using marker gene to define cell types. So I guess one challenge we are facing is how do we trust, how much confidence do we have in RNA-defined cell types? Is there a gold standard to do that? And is that a question that we should address here?
Well, and you can't always decide a cell type, right? It is true, it's nice when there are different algorithms out there for trying to set up clusters, but first of all, they don't always get the same thing. Second, they can, where do you draw a cluster line is not always trivial.
It's, it's actually, Joe can say that, but in the BRAIN initiative, when you read papers, some people define 500 BRAIN cell types. Others define just fewer, 100 or 200. And who's right, who's wrong? Or is there a right or a wrong answer? I don't really know. It depends on...
Concordance between replicates is good, but a lot of times groups don't do replicates. So, I think it's still an open question as to the lumping versus splitting issue.
Yeah, I mean, I think, one kind of strategy is taking a hierarchical approach where you kind of have your, like in the brain, neuronal versus non-neuronal is kind of the biggest split, and then you can go more and more granular. And you can do that with any tissue in terms of the cell type composition, but you've got, big groups of cell types that then feed into cell types. But then, when you start getting into the more rare, granular populations, I think then it's just a semantic argument as to whether it's a cell type or a state or, which, I don't know if it's good to argue about that as opposed to just kind of saying, here's the level of granularity, pick what you want, and then for whatever analysis you want to do.
Hi, Mike Payson. I think you raised your hand, but you're muted, Mike. Can you turn on your mic?
That hierarchical presentation could help balancing the information presentation to specialists in the field and people further away from the field. And as people are less invested in the particular biological problem, that higher level visualization of what's going on may be all they need. And people that are actually working on those individual cell fates and states may say, no, I actually want to drill down and see what type of activation has this particular cell fate experienced.
I think this is a question not only is related to semantics, but also related to technologies. We, for example, know that RNA is quite dynamic. Even for the same cell type, it could be changing, depends on the phase of the cell cycle, depends on the physiological context the cell is experiencing. So is RNA the right or best modality to define cell types? I guess the answer is open, right? The jury is still out. Now, with the context of multimodal profiling, can the use of other modalities such as DNA methylation, chromatin openness aid the definition of cell types? I think that there's a real possibility where this can help, right? And that's where probably computational analysis or tools should be developed to make it happen.
Well, it comes down to what's a cell type, right?
Yeah, it's a circular, I agree, a circular argument.
Finding a defined entity of some sort, as a heat shock cell, cell type from a non-heat shock cell.
That's right. In my view, a
same cell, but in different states. Most people would probably say.
That's right. That's right. In my view, different cell cycle phases should not change the cell type, but they do change the expression, right? So there's cell state versus cell type. I think therefore maybe what we could argue would be define both the cell type and the different state of the cell of the same type, right? If that can be done. And I would say also that epigenome is regarded as another way to define cell types, right? We do know that DNA methylation is a relatively stable mark and is variable across different cell types. And that's where potentially by simultaneously profiling RNA and DNA methylation that would define both cell type and the states.
Not to mention age.
Yes.
Joe, you want to weigh in on that?
Yeah, I think we see if you cluster methylation and RNA from the same nucleus, you get different clusters, right? There's different information. You get overlapping clusters, but there's some that are unique that are being driven by the methylation and others that are being driven by the transient state of the RNA. So,
and are they different cell types or are they different cell states?
No. I would say, I would agree with what Bing said, is that the dynamics are very different. If the RNA, so I put circadian rhythms into that transient state, just like cell cycle is in that state. That you see changes that you can assign to some environmental component, whether it's time of day or perturbation, et cetera, but that those things don't change as much at least that we see in the methylation data.
So when you do cluster analysis on methylation, do you get better separation of cells? Is that?
Well, if you're looking at if you're comparing head to head, you can't do as many cells. Andrew can tell you, maybe he's been working on methods to get very much higher numbers, but you get very good resolution from a small number of cells, but you lose the rare ones because you just don't sample them as much.
Right. Okay.
Yeah, so in that regard, ATAC-seq data or open chromatin data also is probably in between. For the same cell types, it's probably not changing very much, but I mean, it's probably more dynamic than DNA methylation, but less dynamic than RNA.
Well, except you can get many more configurations of open chromatin, seems to me, than you do for RNA. It seems like when you have genes, you can actually better stratify them with the ATAC data. From cell types defined by RNA, you can subset out with the ATAC data, right?
Depends on the cell type, though. We have been analyzing some brain cell types. And what we are finding is that we can actually, for some neuronal cell types, we can better separate the different subtypes with ATAC-seq data than RNA-seq data. That's just anecdotal observation.
So then to generalize this, maybe stepping back a minute, the question is, what assays are best for defining cell types? Okay, first bullet, I guess I got to get back to Google Doc there. What assays are best for that? And then how do you decide who's right?
I mean, as we said before, that could go either way in terms of, ATAC to RNA or RNA to ATAC, which one's going to have more granularity. But I think getting at what Bing and Joe brought up as well with just the kind of how stable these marks are, right? So, with methylation being kind of the most stable, that may be more cell type, whereas if you're shifting to more, RNA that can change much more rapidly, it's more cell state. Again, it's starting to get into semantics, but, it could be, where it falls on the hierarchy, and, that might be different cell type to cell type too.
Well, usually at the end of the day, it's when you superimpose a biological meaning on top of that. So when you see some differences, so you can say, well, that's due to circadian rhythm. It's a time of day, or it's an age, or it's something.
Yeah.
So it's usually then how you can, so when you ultimately come up with an explanation, that lets you define it, I suppose, in some simplistic terms.
So again, here, how do we separate cell type versus state? Is there a agreement among the community or folks practicing this art where the line, when do you say you have become a different cell type? When do you say you're in a different state?
I don't think many groups have done sort of, I mean, one way to do this is sort of developmental time. Many of the samples that I've seen are sort of static, you know, it's like liver at this point, lung at this point. I think you'd get some of that information if you had more developmental trajectories, because then you could see, you know, if there's a small population of cells that are kind of, you know, if there's a normal development and then you see that there's a small population of cells that are actually doing that later in adult development, you can say, oh, those are progenitors of et cetera. So I think we're really stuck in, at least for some of these analyses that are based on very static time points. And I think you can get, yeah, I mean, you can always, in systems that are very dynamic, you have pseudotime and you can say maybe that's a state, it's transitioning. But for a lot of the other tissues, at least the data I've seen, you know, it's really just one or two stages of development. So I think it's harder to, I think once you get more information, you may be able to assign what's stable and what's, you know, sort of dynamic.
Mike Payson,
I think for people that are studying the biology of specific cells, sometimes states are things that are reversible, whereas fates are not. So we're talking about circadian rhythms, changes in cell cycle, or lymphocytes can become activated and then go back to a resting state. Whereas if you go from a progenitor to a terminally differentiated cell, that's a change in cell fate, but that's when you've already been studying that cell type for years and know those answers.
So basically, Mike's point is, if you define a type, you don't expect one type suddenly change to another without, say, reprogramming efforts. But with first state, there is interchangeability between them. Is that the definition we can adopt for the practical purpose? That seemed like a pretty good test, right?
And the problem is that you don't have those two at the same time in the same cell, right? So you have the differentiated state maybe in one cell and something different in another cell, because you're not measuring the changes in that cell over time. You're just measuring the final state, or you see other cells that look like they're in transition. What are they? Are they your unique cell type, or are they, you know, undergoing some environmental exposure, you know, like clock or timing, etc. So if you had, you know, sort of recording of a cell over time, right? That's maybe another session. That could maybe tell you what's a stable state versus a transition, because you're measuring both. Whereas if you destroy the cells, then you have a problem. You can't actually measure both things.
Yeah. And this is the test that probably could be used to validate the definition of type. But computationally, it's hard to implement, right?
Yeah, and I like to mention that we have been looking a little bit into this in terms of generating single cell RNA-seq, single cell ATAC-seq, and also the joint from the same set of cells, single cell RNA-seq, single cell ATAC-seq. But I find that either using either or RNA or ATAC-seq, you can get those broad cell classifications, right? But what's really of interest is when you get into the point of, say, T cells, and you want to further take a deep dive into it, you look at the transcription, and you might not be able to separate all the subsets of T cells from that. So then the multimodality becomes really interesting because from the DNA, say DNA methylation or the chromatin accessibility, you could further tease out the resolution of clusters within reclustering, say a broad cell type, right? So say you have your myeloid cells, you zoom into those, then you can recluster based on the attack leak data that you have, or epigenomic, say methylation side of it. And that's when it becomes really quite relevant. But even for myself, I don't know whether there really are other methods that can push the envelope to further integrate and define, as you're mentioning, not just the cell type, but differentiate from cell states. For the RNA-seq side, of course, you can use the pseudo time, and really nicely, if you have different time points, you can follow that through. But I agree, that's also a limitation. But I think there's two aspects, right? So there's like the broad cell type classification, which either or would do, and then the further fine tuning of the broad cell types that you're looking at. I think that's where there's a lot of room for improvement.
So perhaps Joe can say a little bit more on the experience of Bring Initiative. Do you see, do you try to integrate methylation with RNA, single-cell RNA-seq data, and detect a difference between the type and the states, or not?
So there's a paper on the bioRxiv that was posited a couple of weeks ago that does a multimodal integration of methylation RNA of various types and attack, as you know, for motor cortex. I don't think that specific question was addressed. Basically, there's been a reliance because the RNA data seems to suggest, you know, that there are these other states that everything gets mapped into the RNA data, whether or not those are, so you might have one cluster of methylation or one cluster of accessible chromatin sites, and that'll map across 4 different RNA clusters or so. But it's not clear whether that's real or imagined, you know. So I think you need other kinds of experiments to say, as someone pointed out, sort of functional experiments.
Mm-hmm. Well, if the RNA suggests different functions, then you, again, if there's, it usually comes down to putting a little biology around the differences, and that usually winds up defining what's going on either at a state level or a cell type level.
Yeah.
At least that's how the human mind works.
So it's kind of, you know, you could think of it in different categories, I guess. There's, you know, cell types that can actually be identified across all modalities, right? You know, broader classifications where no matter what assay you're looking at, whatever modalities you're looking at, you're going to be able to get these sort of classifications that's, you know, shared across all of them. Whereas other properties or other, you know, cell subtypes or types might be only specific to one modality. And then I agree, you need to have some biological function regardless to actually tease those out, you know, validate them in some way. But, you know, just in terms of framing the problem, I think, you know, kind of splitting it into those 2 categories, I think makes sense to me.
Yeah, me too. I mean, if you think about ultimately when we start doing single cell metabolomics, I know the Vanderbilt group's doing spatial metabolomics information, right? Just think about it. There's lots of different metabolically active states that are possibilities for cells. And so, again, I would think most of those are states, but they're still worth describing in some entity.
So what would be needed for us to move this to the next stage? I think we all recognize the challenges, and part of that is semantics, part of that is technology, but how do we make concrete progress as a group?
Well, this is probably the kind of thing where a team needs to form together and make some operational definitions of what we're describing here and get buy-in from a larger group. And I think to the extent that could be done, that would be useful for going across consortia so that the different groups are all talking to each other on the same terms. Seems to me that's the goal of what we're trying to do with the blue box.
Yeah, yes, that's a concrete steps that we can take. What kind of specific expertise we need to be put on this team? I guess we many of us on this call have wet lab experience generating the data and analyze them.
You need rock'em sock'em computational folks. Definitely not me. You need people comfortable with different assays. You know, that can actually tease out different cell types or states. So I think you need a combination of experimentals, computational folks. And I mean, I think there's also some very interesting statistical tools people are trying to use to address this problem. So I think getting some of those folks on would be quite interesting as well. Guys like Rob Tishrani probably, you know, we used to set up clustering for microarrays. He'd be a great guy to bring into this kind of thing. And I guess Bong Long from my lab's very interested in this sort of stuff. So anyway, lots of different kinds. Mike looks like he wants to talk.
I think you'd want people with expertise in the biology of these cells that know the different fates and the states and what the cells do, so that they could then help interpret, well, if this gene has changed in expression, I know what that means, or this other change, I don't know how to interpret it.
Yeah, I agree. Probably identify a biological system as well for tackling such problem. Let me move on to potential members of such a working group. I just heard Mike nominated Meng to join. Is that right?
Well, she's my postdoc, so I'll let her decide. Certainly would nominate Anshul. Will Greenleaf. I'll nominate lots of people who aren't on this call. Anshul was, I don't know where he is now. So, because they do this stuff for a living, they would be good. I mentioned Rob Tishrani. I don't know if he'd be interested or not. He's a statistician at Stanford, did a lot with clustering.
How do you spell his name?
Tibshirani, T-I-B-S. Oh, I don't know. I'll send it to you in an email there. But Mong, if she's willing to, she's a postdoc, so she's a statistician. So
great.
If she agrees, great.
Yeah, I will be happy to work on it.
So Jill, do you have someone your lab who might be interested in taking the methylome angle?
Yeah, I'll send a link to the recording. I guess it'll be a recording and they can listen to it and then ask for a volunteer from our lab.
Mm-hmm.
Maybe somebody that understands also the experimental aspect with it. From the morning session, we have some overlap with what you're discussing. It was mainly more related to the multimodal sort of quality control, that really sort of understanding how the different data type, what the problems are that are unique to the multimodal approaches. But yeah, I can try to nominate somebody from the group that would be interested in this.
I'll nominate my lab member to also join this discussion. Do we have other volunteers on this?
Yeah, this is Benedict Anchang. I would probably be interested.
Okay, great. Anyone else who's interested in this topic? Let me maybe make us again comments on biology. I think Mike Payson's suggestion is absolutely dead on target. there, among all the biological systems that we could potentially look at, I would highly recommend brain initiative data. As Joe mentioned earlier, the brain initiative has generated all kinds of single cell modality data. We heard about single cell RNAs, MERFISH, single cell attack, single cell methylation, and also patch-seq, and all kinds of others. The most important thing is they have been generated for a common brain region, the primary motor cortex. And there is a bioRxiv paper that described this effort that was posted a few weeks ago. This potentially could save a lot of effort from data generation side and to facilitate the computational analysis. So I certainly felt if we want to move this forward, that may be a good biological system to focus on. And in that regard, we need someone who has deep knowledge about brain anatomy or neuroscience. Would you say? What do you think?
I have a question. This is Benedict. Is the system supposed to be under the static, or are we studying the cell types or the states within a dynamic system? Like if we
Good question. Yeah, the brain data is static. It's not a developmental context. So that would be the drawback.
Because if you want to differentiate between state and cell types, I think we need a system that can capture both static features as well as dynamic features.
That's right. And what would you recommend for that purpose?
I have been thinking, like, we recently published a paper with Rob Tibshirani on like plasticity of cells, like EMT. Those kind of systems are good because you can capture the entire from the beginning to the end of a process. And that also has some kind of an internal control, because in that sense, even if you could computationally or experimentally determine the cell types, you know that they are within a context of the entire process. So I don't know if this, but I think this is a way to move forward if we really want to kind of capture or validate cell types and cell states.
Good point. Hematopoietic stem cell system, right?
Yes, or yeah, systems that also have the reversible nature of things as well, yeah.
So did you want to take on the other blue? When do we end on this session now that we're all confused about timing?
We have
take on the other blue box. I don't know.
Do we have 70 minutes? I don't know.
Good question.
So we started 2:55. So
it was 75 minutes?
No, it's about 40. It's about 45 minutes now.
Oh, we have plenty of time then. Okay.
We have 30 minutes, right? So we have 20 to 30 minutes, depends on how we want to proceed. So I just want to complete the box one, and I know I'm trying to fill it. The first one, we defined the problems where we can collaborate on, and we made some recommendations on steps moving forward. We identified some potential members of this working group, and we were discussing potential biological systems or model systems where this problem can be explored. And I think Benedict,
by the way, there's a group that's working on this. I think it's part of H10 that Dana Pear is leading. I think it's built around this problem.
So do you think Dana Pear should be reached out, right?
I would think so.
Okay. Yeah, I also felt hematopoietic differentiation might be a good idea. What else? Do we have other suggestions for this? We can always come back and visit and discuss this in the context of a working group. Okay, so in terms of email contacts, who would like to be a leader for this working group? Do we have any volunteer? That we can put on the email contacts?
I could propose myself, Benedict.
All right, great. Why don't we put your, do you have access to the Google Doc? Can you put your name, email address there? We need at least one, but if we have a second volunteer, that would be also great.
Let me see.
So, so while Benedict putting your contact information there, I think we kind of wrap up on the first box. And this box is really about how to leverage the multimodality profiling data to better define cell types. We could now we actually have some actual data to, actual time to entertain the second question that we discussed about noise. Okay, so again, let me try to describe this problem, define it a little bit better. How to count for noises in the data sets, that's probably not the best way. Maybe what we can say is, what are the sources of noises in our data? And what can be accounted for? Right.
Well, I forgot who brought up. Was it Simon or Andrew or somebody?
From multiple people. One is, I think Sebastian from UCSD did mention that this is a challenge that we faced when we deal with, for example, barcode collision. But Andrew also mentioned that there could be a lot of noises also. This was a topic maybe even brought up in the morning session.
Missing rare cell types.
Yeah.
Also, people have different lysis protocols, so they don't quite capture necessarily the same cells. Sorry, cut you off, Simon. Go ahead.
Oh, that's fine. It's just the missingness in the data, like you were saying. So I think there's a couple of things. It's, you know, the multiplets, the barcode clashing, as well as once you have the data, the missingness in the data.
But another thing is like by ambient RNA molecules, for example, in the single nuclear RNA-seq, where you have like some genes which are very highly expressed in one cell type and might tend to stick to nuclei from other cell types.
Yeah.
And same with chromatin as well. I mean, you can get fragments of chromatin that rupture and then get stuck to other nuclei. And I mean, and I think it's universal across generally all platforms, whether you're doing droplets because they co-encapsulate or combinatorial indexing where things might stick together. And I think it is very different depending on the modalities, you know, in terms of what buffers you use. It might prevent ambient RNA, but might create more ambient chromatin and vice versa. So.
Yeah, so other sources that we could name them. I guess when we're dealing with 10x data, this ambient RNA contamination issue is definitely there, and the same is for fragments. So this is not a unique issue for certain platforms. Among all these sources of errors, what should we address as our top priority?
Mike Pazin had his hand up, by the way.
Oh, hello. Hi, Mike.
Yeah, I don't know if this is more a metadata issue, and somebody had this in the chat, but at a larger scale, if you sample the same tissue or organ in different places, you'll get variability. You know, if you think of skin from different anatomical locations, it's different. So, you know, how do you know that somebody looking at skin is looking at the same type of skin or the same part of a liver or the same part of a bone?
So heterogeneity within a tissue, and then of course you're circling across people, you have that same issue you're dealing with.
So I think just to add to what you guys are saying, I think we have to, it's a trade-off between within and between variability and noise. Which one we want to sacrifice or trade off? Because they kind of like affect the entire process of the biology itself, depending on when we integrate the data.
We were just dealing with more about two sources of errors, but primarily systematic biases that are not due to biological variabilities, rather due to the assay platform, perfectness of the assay platforms. I guess that's what we discuss as noise. Okay. So when we have a good idea about the systematic biases, then we have a better handle on biological variabilities. But I think we probably here want to prioritize systematic biases and how to remove them. We do want to detect biological variabilities, and we want to remove them. But I have to say that some methods such as Doublet tend to remove biological, overcompensate and at the expense of biological variability. So that's something that we need to be watching out for.
Is there a good way to discern transitional states from barcode collisions or doublets?
That's a very technical question, maybe for some of the computational folks.
So I was wondering if that could also be a challenge that could be addressed.
Yeah.
I mean, you're talking about, it depends on the platform, or is it across all platforms?
Across all platforms, because you could think like a transitional state might be a mixture of two cell types.
Yeah.
So that shows up as a mixture of two cell types.
But I think for antibody-based, it's easily solved at the pre-processing stage. But I don't know if for like RNA-seq, yeah, I think that would be an issue for those kind of technologies.
So Sebastian, I think the question you ask is an important one, but probably in the context of Scrublet, that's a relevant question. I'm not sure that's the only way we deal with barcode collision. And for many other source of errors, potentially it's better to look at how the errors were generated and how to computationally compensate for that. Yeah, but certainly the existence of such intermediate cells can be explained by either real biology of intermediate states or by barcode collision or by contamination. So that's a sign. That doesn't mean it's noise. It could be just real biology, but what we probably want to find out is what's the best way to identify noise or errors.
Also, I want to comment on the fact that recently they have also been shown that there's this dormancy period between that genes just go dormant, and those two, I don't think we are able to capture that as well in terms of noise. So it's trivial because you have these periods of dormancy between that genes just go, and that is also challenging in terms of controlling for other things like doublets and everything else. That's it. It's something that was recently just observed. It's most likely one of the contributors of those zeros and things like that.
Benedict, can you say a little bit more clearly? I'm not sure I quite get it. When genes go dormant, how does that contribute to noise?
No, I'm saying that the dormancy period, because typically we tend to model by modality, but it could be that during periods of dormancy, the genes that, so it's not that it could contribute to false positives, but which are really true biology, but then they could add. So those are things that it's also difficult in terms of processing or developing methods for clustering and things like that.
So I'm here want to provide some way to move forward. One way I'm thinking is how do we, we might need to define accurately the sensitivity and the specificity of each assay platform that we're dealing with. I know this is probably this question that would apply for both single modality or multi-modality profiles, but we probably, if we need gold standards to define such assays, sensitivity, and only then we can basically determine what is noise and how likely we are observing noise versus just a biological variability. And I'd like to comment on that. Platforms such as Murfish is known to have very high sensitivity and detection efficiency compared to, say, single-cell RNA-seq type of platforms. And that's, I guess, for different technology platforms, better definition of the sensitivity, specificity would be needed for us to move forward on this.
I think another interesting question too in that vein is, depending on the modality, some modalities might be more sensitive to, or it might be easier to identify doublets or noise in some modalities versus others, just based on their inherent nature, dynamic range, and kind of teasing that out. And potentially, if you do a multimodal assay, you could, target one of those properties, depending on whichever one seems the best for discriminating doublets and identifying those, use that one for that process and not the other, as a way of using the modality and how they inherently behave in terms of noise for that purpose.
Yes, I think at least in our hands, the RNA-seq and ATAC-seq, if you try to assay them together, the sensitivity is probably not as good as single modality. It's coming close, but it's going to certainly suffer. But so that what that means is you're detecting 5 or 10% of one modality and 5 to 10% of another modality, and if you try to integrate the two together, you have to take into consideration of the high false negative rate and potentially modest false positive rate for each modality from the same cell, right? Mm-hmm. So that will definitely present challenges in integrating them. So this goes to computational folks to say, how do you properly assay these two modalities from the same cell?
It's probably just an observation that we've had, and it may not be accurate, but certainly the ambient RNA, it doesn't cluster as well as specific cell type. And if you look for enrichment in particular pathways and things like that, it doesn't seem to enrich for any specific pathway. It seems to represent all. I don't know in the methods of removing ambient rather than using objective or subjective ways, whether there's any way that you can include those sort of information.
Anyone on this group who is experienced in that? Does anyone have a specific solution to remove ambient contaminant RNA?
So the context that we typically see ambient RNA is not like a single cell barcode is just comprised of ambient RNA or ambient chromatin. It's more, we're doing barnyard experiments, actual on real tissue as opposed to cell lines, which are a very idealized form of a barnyard. And we start to see, cross-cell contamination at various levels, depending on often the quality of the tissue and how it's been preserved and things like that. So, and to actually identify that in a context where you're not doing a barnyard experiment, we don't have any good ways of doing it. I mean, we'll see if it's an experiment with a lot of ambient chromatin, we might see, sets of cells that are clearly showing markers for, excitatory neurons, but there might be some signal at genes that are for, like specific to oligodendrocytes or something, for example. But it's much, much lower signal. So it's likely ambient chromatin. So actually quantifying that without really having high quality profiles already, which is what you're often trying to do with an Atlas project, is very difficult. And I don't really know the path forward on that.
All right, I'm sensing that we are heading on a stage where we probably should try to wrap up the discussion. I'm trying, let me try to identify what are the key challenges in this topic. There are many sources of errors in the multimodality profiles. Ambient RNA or nuclear fragments that stuck to the cells could be a major challenge. Missing cell types, barcode collisions, and the other type of source of errors are known. So the question is, can we computationally identify and remove such errors, right? So that's the question we're dealing with. So of course, depending on the source of error, you have to come up with different ways to remove, identify, remove them. One way that have been proposed in the field is to look for signs where you have two very different markers to be present from the same cell. And that would be consistent with barcode collision or contaminants, but they can also be interpreted as some kind of intermediate cell state. So we don't know how to address them yet. So that's one challenge that we are facing. So I don't know whether that fully captured the discussion, but that's how I view what we have heard. So to move forward, how do we bring this discussion to another stage with actionable items?
I think we need a good system.
With general applicability, right? But we would like to have a leader first, whoever felt this problem is sufficiently exciting, please step forward and maybe assemble a working group and move this to a stage where we can conclude something. Do we have enough people excited about this?
So it's something I'm interested in terms of, you know, what a working group would do. You know, it's figuring out exactly. So some of these questions would be, you know, better experimental techniques that don't have this noise. You know, and I think really what will be most useful in terms of, you know, cross consortia collaborative efforts would be individuals that are, you know, computational individuals that do this work to try and identify what these, you know, noise aspects are and remove them from the system or filter, you know, cells out or things like that. So I think that would be the best group.
And quality control in general, one could argue, perhaps. Maybe that's getting too broad.
Well, we had that discussion this morning. If you maybe, when we get to the final part where you can look at each one of these blue documents, there's Andrew put together a QC metrics for multimodal assays and in our call this morning. And there's some, and that maybe that some of these things that maybe this topic can be somewhat combined with the.
I agree.
A lot of experimental issues in here, and maybe they can combine with the computational.
And if it's too much, then you break into subgroups.
Yeah,
we'll take it on.
Yeah, I think it could make a lot of sense based on the conversations earlier to bring these together, because it is very much a QC component in the conversation we've had now. So yeah, I think that could work out well.
That sounds like a very synergistic discussion. I agree. This probably should be discussed in this context of both experimental and computational assays.
Try and find various sources of error.
Great.
And set up solutions.
Yeah. So in terms of recommendations, should we just recommend that this problem being addressed together in the morning session? In the breakout session of the multimodal tool develop session. Did you have 2 or 3 questions in that? What, this is the one?
I think we had 2.
We had two.
Yeah. Had 2, yeah.
One was developing QC metrics specific to multimodal assays.
Oh.
Recommendations were, you know, QC standards for different sequencing-based readouts. Data standards for the single modalities are established, and then how do you modify those for multi-omic assays to compare them? Assess artifacts induced by cell dissociation or other methods, create a set of genes that can be associated with different problems. So if you've got, you know, like basically lists of genes, like if you have this in that sample, then you're likely to have some issue with contamination. So maybe gene lists would be helpful across cell type contamination, blah, blah. From actual samples, Andrew mentioned that. If you're doing these sort of barnyard experiments with cell lines, it doesn't really capture issues with tissues.
You got 1 minute, guys. So why don't you say, work with that other group on these issues that Dina's just listed above the line.
Yep.
Form a working group.
I can request that blue box and this blue box be co-located on the virtual wall so they can be together, and then you guys can figure that out a little bit later.
Cool. Thank you very much, Shannon.
Yep.
I thought we did a great job concluding this session, and thanks everyone for participating.